IB Bio student. So today we are going to continue with the structure of tRNA and how tRNA gets activated. So last time you all looked at the structure of the ribosome, you saw how it has a small subunit and then a large subunit. So remember that this is all within the process of translation, right? So let me get my pen out. So if you were to look at this ribosome, right, and you were to explain how translation happens, well, you know that, oops, pen there. You know that mRNA gets translocated on the ribosome, right? And then tRNA, and I can't draw, so don't laugh at my drawing, tRNA then comes in and it brings an amino acid that attaches to these different sites within the ribosome, right? Remember, we call them the EPA site. So tRNA comes in, gets activated, then the bond forms in the peptidyl site, and then it exits. And many tRNAs come and undergo this process, and that's what allows for the polypeptide chain to grow. Or we call this a chain of amino acids, which is also a polypeptide, which is also a protein. Okay, that's like translation in 2 seconds there. So as we move along here, we're supposed to recognize the structure of transfer RNA. And notice that we can look at it in a flat secondary structure. We could look at it in a more three-dimensional tertiary structure. Or we can also look at it as a space fill model. Okay? Either way it's shown to you, you should be able to recognize it. And the way I recognize it is because in these tertiary and space filling model, this acceptor stem kind of like, sticks out like this, like a little stem, right? And it doesn't really show you here the shape of an actual T, right? But it does here in the secondary structure. So that's another way we can recognize this as tRNA. So you're supposed to recognize the clover leaf structure, and then you're supposed to recognize the 4 regions. So the first region that you are to recognize is the acceptor stem region. And this is on the 3' end of the tRNA molecule. So think of tRNA as an mRNA that is folded. Okay, so notice that there's interaction here between the bases of this tRNA, A, U, G, C. So here we see that the acceptor stem is on the 3' end, and then that is identified by having these bases, CCA. Okay, and in this side of the acceptor stem, this is where the amino acid is going to attach. So remember that I told you that as each transfer RNA brings an amino acid, the amino acid chain is going to grow, and that's how you're going to end up with a very long protein or very short protein, whatever protein it is you need to make. The other part of the tRNA that you are to recognize is the anticodon. And you probably remember this from last class. But the anticodon is this tip of the tRNA. And it's made up of 1, 2, 3 different bases. Now remember that these are the bases that are going to attach to the codon on mRNA. So remember that if this were mRNA, mRNA would be read as a triplet code that we otherwise call a codon. So the codon on mRNA pairs up with the anticodon on tRNA. Okay. The other part that you're supposed to recognize is the T-arm. So that's this region right here. And this is the region that is going to associate with the ribosome and is going to associate with the EPA binding sites. So first this arm is going to attach to the A site, and then it's going to attach to the P site, and that's what's going to induce the peptide bond to form. And then it's going to attach to, actually not attach, but just move on to the E site, and that's where it's going to leave. Okay. And then we have the D arm, And again, you probably never heard of the D-arm, but the D-arm associates with the tRNA activating enzyme. And this is the enzyme that's actually responsible for the catalytic function of adding or attaching the amino acid that goes on this acceptor stem. Okay? Now, this tRNA activating enzyme has a very big name. It's called Acyl tRNA Synthetase. And I don't think you're responsible for knowing the name. It's not in the application, but I'm going to give you the name anyways. Okay, there it is. Okay, so let's move right along then. So how does that tRNA get activated? And if I'm going too fast, remember you can stop this video and pause it, watch it again. All right, so how does tRNA get activated? It does so by the tRNA activating enzyme that I just mentioned, right? So remember that enzymes and substrates have a specificity. They fit like a lock and a key. And not really, it's more like an induced fit, we learned, right? And then by the role of phosphorylation. So if you remember from last year, phosphorylation is when ATP, and ATP stands for adenosine triphosphate, it's when ATP, who has 3 phosphates here, loses 2. So if you lose 2 phosphate groups, right, you are going to phosphorylate, and then you are going to provide the energy necessary for the enzyme substrate complex to catalyze and do whatever it needs to do. Okay, so that's what phosphorylation is. So I think the easiest way to understand tRNA activation is just actually look at this diagram. It's very helpful. So here we have the enzyme. Okay, this is tRNA activating enzyme, and I also gave you the name of this before. I said that it was called Acyl tRNA Synthetase. And the reason why it's called Acyl tRNA Synthetase, where all enzymes end with ase. But the reason why we call it Acyl is because this is the enzyme that's going to be responsible for binding the amino acid. Now, remember that there are, on average, there's a little bit more, but on average, 20 amino acids, right, out there. So if you think about it, there's got to be around 20 different amino acyl tRNA synthetase, depending on which amino acid you're trying to bind. So that's what they mean by this. Each amino acid is recognized by a specific enzyme, although the enzyme may recognize multiple tRNA molecules due to degeneracy. And this is because, remember that in the codon table and in the code, there could be multiple codons for one particular amino acid, and that's what that means. All right. So what happens afterwards? So here we have the tRNA activating enzyme. The enzyme binds to ATP. Here's ATP, adenosine triphosphate. And it also binds to a specific amino acid. So remember, it's got to be specific because of the mRNA tRNA complementary codon to anticodon pair. So it's going to bring a specific amino acid. So here we see, once they bind, you form an amino acid AMP complex, an amino acid AMP complex. That's because this is no longer ATP. This is now AMP, okay? Because it's lost 2 of its phosphate groups, and now it's only got 1. You see that little yellow circle there? So now the amino acid and the AMP are connected. Do you see how they're connected by that little line right there? So now they're connected. So now it's a complex. So now that it's a complex, that means that the energy is there to recruit that specific tRNA molecule. So tRNA is like, all right, you're ready to go. You're phosphorylated. So the tRNA is then comes in to a particular active site within the enzyme. So this is what we mean by enzyme substrate specificity. And notice that there's different sites, right, for this enzyme substrate specificity. There's a site for the amino acid, there's a site for the ATP, and there's a site for tRNA, which is not something that you've probably seen before in an enzyme substrate complex, but FYI, there's more than one active site. So now the tRNA is bound to the amino acid and the AMP is released. Okay? So now that the AMP is released, that means that this enzyme can say, goodbye, tRNA. You are now charged. And what does that mean? That means that the amino acid is now attached to the tRNA. And now what happens to this tRNA? It goes and it finds the ribosome. And remember that there's two subunits of the ribosome. It finds the ribosome. The mRNA is translocated on the ribosome. It finds the EPA region on the ribosome, and then it attaches to the A region first, moves to the P region, forms the peptide bond between that amino acid and another amino acid that might be growing, and then exits. And then this tRNA can be reused again because this process can be repeated, right? Remember that enzymes can be used multiple times. Okay, so now tRNA is activated or has been activated, and translation can happen. Okay, that's it for today.